Hello friends, Ace Botany, next episode. And in this episode, we'll be discussing photosynthesis, fifth one. Now remaining one more that we can finish in next episode. So we can continue with the photosynthesis, fifth one. So for convenience, I will share the screen, then we can proceed. I think screen came. So shall we proceed? So photosynthesis in higher plants, fifth one. What all topics we'll be discussing in this episode? Basically, 2 plus an additional cycle. After that, questions, 5 theory, 5 NEET model with the key. So we can proceed. Starting with the first cycle in this episode, that is C2 cycle, otherwise called photorespiration. Again, another synonym, oxidative photosynthetic carbon cycle. Another one is PCO cycle. So many names are there for C2 cycle. And its discovery and initial work is concerned with the famous two scientists, Dicker and Teo. Worked in tobacco in around 1959. Now, what is the meaning of the term photorespiration? Photo already we know because photosynthesis, that is heading of this chapter. So photo means light. Respiration means taking of oxygen and giving out carbon dioxide. And in which all plants this is commonly seen? It is commonly seen in C3 plants because it is an adaptation that we'll be discussing later. It is a cyclic pathway. And this photorespiration is a kind of respiration seen in photosynthetic cells only. I think up to that introduction is clear to all of you. And the main enzyme, it is Rubisco for both the carboxylation, then the enzyme ending will be carboxylase, and oxygenation. Then the ending of the CO will be oxygenase. And under which conditions water respiration will be normally taking place? Mainly C3 plants. 1. High light intensity, hot and dry environment, limited water supply, high oxygen concentration, comparatively low amount of carbon dioxide. And practically when we think, it is a wasteful process. Why? No synthesis of ATP, no synthesis of NADPH, no synthesis of sugar. And besides this, there is loss of 25% of the fixed carbon dioxide from mitochondria. So huge loss of carbon dioxide. And which are the cell organs involved? Mainly 3 cell organs: chloroplast, peroxisome, mitochondria. Of which peroxisome, you know, it is a microbody. Other 2, comparatively double membrane cell organ, chloroplast and mitochondria. And what is the need for this C2 cycle, photorespiration? It is to protect chloroplast from photo damage, excessive heat. And it helps to dissipate or dissipation of energy after closing of stoma during daytime due to water stress. Photorespiration takes place in C3 plants, not in C4 plants. And a diagrammatic representation is given. I think first page is clear. Second page, if you observe, you can find a diagrammatic representation. This is the representation. You can find 3 cell organ, chloroplast, peroxisome, mitochondria. Even carbon numbers I wrote there. So starting with the RUBP, reaction first is in chloroplast, couples with oxygen, photorespiration. Enzyme is oxygenase. Here you can find. And the first product formed in chloroplast is phosphoglycolate. It is a 2-carbon compound, hence the name C2 cycle. Phosphoglycolate will be converted into glycolate. Again, 2-carbon compound. Glycolate will be shifting into peroxisome. See, again couples with oxygen, second coupling with oxygen. It converts into glyoxylate. Cell organ is peroxisome. Glyoxylate is converted into glycine, simplest amino acid. So glycine first formed in peroxisome, 2-carbon. Now glycine will be shifted to mitochondria. Glycine is a 2-carbon compound, simplest amino acid. So 2 glycine has to involve. So 4-carbon, from which carbon dioxide will be removed. So it again becomes 3-carbon. It is another amino acid called serine. So serine formed in mitochondria. Now in mitochondria, 3 famous sets of reaction takes place. Our popular question. One is deamination here. One is decarboxylation here. And one is dehydrogenation here. So 3 sets of reaction takes place in the mitochondria in connection with the C2 cycle. Important question. Now we have serine entered into peroxisome. It will be converted into hydroxypyruvate. Here you can find hydroxypyruvate, again reduction, that type of reaction here. Then hydroxypyruvate will be converted to glycerate. Glycerate becomes 3 carbon. Then from glycerate, that is shifted to chloroplast. ATP consumption, etc. That much details not needed in NCERT still. And it gives rise to PGA. That means it is linked with the, connected with the C3 cycle. So that way it proceeds. So 3 cell organs, chloroplast, peroxisome, mitochondria, and 2 amino acid, that is glycine and serine. Glycine formed in peroxisome, serine formed in mitochondria. And that way the reaction proceeds. So chloroplast, peroxisome, mitochondria. I think idea is clear for C2 cycle. Now we can proceed to next cycle, more important, for exam point of view, C4 cycle. With that we can proceed. As usual, many synonyms are there. C4 cycle, you can find many synonyms. Hatch and Slack pathway, HSK pathway, K stands for Kortschak, dicarboxylic acid cycle, etc. Many names are there. And initially C4 cycle, because first stable product is a 4-carbon acid. Previously C2 cycle, first stable product is a 2-carbon chemical. Now, this cycle was connected with 2 Australian physiologists, Hatch and Slack, 1965-67 time or years. And initial work is connected with Kortschak and its group in sugarcane. I think that idea is clear. Now, in which area we can find this type of cycle? Again, this is an adaptation for dry tropical region because nature understood C2 is not much advantageous. Plants from both the monocot and dicots will be showing this cycle. Typical example is maize, sugarcane, sorghum, amaranthus. Plants in C4 is important. Odd one question will be there. And the name C4, everything written there, name C4 is due to first stable product I already told you. That is a 4-carbon compound called oxaloacetic acid, OAA. And C4 plants have double fixation. Carbon dioxide will be fixed 2 times. One is primary fixation in mesophyll cell, secondary fixation in bundle sheath cell. That is a changeover from C2 to C4. I think up to that it is clear. Now, C4 plant, why it is developed to avoid C2 cycle or photorespiration will not take place in C4 plants. Another adaptation. Because C4 plants have a mechanism that will enhance the concentration of carbon dioxide at the site of enzyme. That is how in C4 plants, photorespiration is avoided. I think that idea is clear. Now comes special type of anatomy. That is another area of question. See, for completion of this cycle, 2 areas needed. One is mesophyll cell, other is bundle sheath cell. And in mesophyll cells, first item will be starting. First reaction, that is carbon dioxide. I will put the diagram like this, it will be easy for you. So first reaction, that is carbon dioxide, is initially accepted by a 3-carbon compound called phosphoenol pyruvate. That is why here you cannot observe much chemical names. Here I specifically wrote, so it will be easy. So carbon dioxide, CO2 will be coupling with a 3-carbon acid, phosphoenol pyruvic acid, and that is present in mesophyll. And in this mesophyll cells, we can find enzyme rubisco is absent. So enzyme rubisco is absent in mesophyll cells of C4 plants. And the first reaction, that is carbon dioxide coupling with the phosphoenol pyruvate, is catalyzed by a famous enzyme called pepcase, phosphoenol pyruvate carboxylase. And product what we get is 4-carbon acid called oxaloacetic acid. That is why the name C4 cycle. Now this oxaloacetic acid will be converted into different organic acid. Oxaloacetic acid is 4-carbon, aspartic acid is 4-carbon, malic acid is 4-carbon. So many organic acids are formed in C4 cycle. That is an adaptation that we'll be studying later. And now we reached into malic acid. You can find 4-carbon. So acid from mesophyll cell shifted to bundle sheath cell. malic acid shunt, etc., shifted to bundle sheath cell through specific channels given by plasma-desmata. Another important question. And in bundle sheath cell, now we reached here from mesophyll to bundle sheath cell. 4-carbon acid that will release carbon dioxide. Here you can find carbon dioxide will be released, and that will be proceeding to C3 cycle, carbon dioxide. That is how C3 takes place here. So after releasing carbon dioxide from the 4-carbon acid, becomes 3-carbon chemical. So another acid will be formed. Release carbon dioxide will enter into C3 pathway. I think up to that it is clear. Again, coming into mesophyll cell, here grana is present. So it is called granal chloroplast, area of question. So mesophyll cell, grana present. So that is called granal chloroplast. Now we will be proceeding to bundle sheath cells. Then we can continue. So bundle sheath cell, that is here. This is the enlarged diagram, labeled one. Bundle sheath cells means cells which surround the vascular bundle. That's all, nothing more. But again, an area of question, special features of bundle sheath cell, popular question. We can find several layers will be there around vascular bundle, several layers. Large number of chloroplasts. Absence of grana. Then it is called egg-granal chloroplast. Previously, what we call granule, here it is egg-granal chloroplast. I marked here very clearly. Thick walls makes impervious to gaseous exchange, closely packed, no intercellular space. And this special type of anatomy seen in leaves of C4 plants is called Kranz anatomy. Important question. Kranz anatomy. Characters of Kranz anatomy, entrance question also. I think up to that it is clear. Now, in C4 plants, chloroplast is dimorphism. Grana present, grana absent. And what is the meaning of the term Kranz? Kranz means a crown, halo, a ring, a wreath. These all are the meanings of Kranz, the words. In bundle sheath cell, Rubisco, here you can find Rubisco enzyme is available but lack pepcase. Previous case, just reverse. Same manner in mesophyll cell, Rubisco absent, pepcase present. Now, the 3-carbon acid here will be formed in bundle sheath cells, transported to mesophyll cell, again through plasmodesmata. And it will be converted into phosphoenol pyruvic acid. So again, regenerating PEP by an enzyme phosphoenol pyruvic acid synthetase. There is another name for this enzyme, pyruvate phosphate dikinase, PPDK. Cycle will be completed. So again, we can get PEP. Cycle can be repeated, and PEP synthetase is sensitive to cold temperature. So totality we can observe C3 cycle is seen in C2 pathway as well as C4 pathway. We can find. So common pathway is C3. I think idea is clear. These are the diagrams. This is NCERT diagram with rectangle showing different stages, labeling question. This is bundle sheath cells, etc. enlarged, Kranz anatomy. And this is little bit more expanded from the sense chemicals I wrote there. I think it is clear. Now we will be proceeding to ATP calculation in C4. So when we take C4 only, C4 side only, 2 ATP per carbon dioxide. C4 we know, C3 will take place. So for that C3 part, 3 ATP per carbon dioxide. So generally when we say total ATP in total C4 cycle is 2 plus 3, 5 ATP per carbon dioxide fixed. So to form an exhaust with the 6 carbon or to fix 6 carbon dioxide. For 1 carbon dioxide, 5 ATP. For 6 carbon dioxide, 6 into 5, 30 ATP. How that 30 comes? 18 in C3 part, 12 in C4 part. So total 18 plus 12, 30 ATP in C4 cycle, complete C4 cycle to form an exhaust or to fix 6 carbon dioxide. So very famous question, this one. Sometimes plants names will be given how many ATP, likewise. I think idea clear. Now, importance, disadvantages of C4 pathway. That we can proceed. First is disadvantage of C4. More energy is needed to extra ATP per carbon dioxide fixed. That way 6 carbon dioxide means 6 into 2, 12. That is why 18 plus 12 become 30. Calculations will be there, common. Calculations I don't get perplexed. This is the idea. If you got this, any calculation you can do. Some plant's name will be given. 2 molecules, 3 molecules to exhaust in C4 cycle, how many ATP that we will be putting. This is a basic idea. Popular question. Now, advantages of C4 cycle. Main advantage is photorespiration will not take place in C4 plants. And there is a mechanism that increases the concentration of carbon dioxide at the enzyme site. So no other reaction may take place. C4 plants are so efficient to pick up carbon dioxide, even low concentration due to high affinity of PEP. Efficient management of water by providing concentric arrangement of mesophyll cells results in smaller area in relation to volume for better utilization of available water because temperature will be high. Even they can tolerate excess salts due to presence of many organic acid in connection with osmosis. So we can find C4 plants has got many adaptations to face high temperature, etc. in tropics. Many organic acid that will be connected with the water potential, etc. And 25% loss of carbon dioxide in C2 cycle, remembering that also will be nullified here. So many advantages C4 plants got except extra ATP. Now comes last cycle, not in direct syllabus, but just mentioning CAM cycle, crassulation acid metabolism, commonly called CAM plants. Crassulation, that is the name of a family, Crassulaceae, cactus, seen in dry habitats. And remember, CAM plants are not C4 plants, they are different. Some reaction similarities may be there, but these two sets of plants are totally different. We can find in plants, total plants, angiosperm when we take 2 types of stomata. That part presently not in our syllabus. Previously it was the transport in plants, but presently that chapter is eliminated. One is stomata photoactive. Photo means light. During light, it will be active. Second type is photoactive. Photo means night. Stomata will be open in night time. In CAM plants, it is photoactive, and nighttime maximum carbon dioxide will be absorbed, and it is stored as malic acid or carbon acid. So malic acid, carbon dioxide will be kept. Daytime, what the event is, temperature is high, so stomata will be closed. So no chance to get carbon dioxide. So what nature provided, from malic acid, carbon dioxide is released, and that will continue for the production of food without opening stomata. So advantage is water will be preserved. And in CAM plants, mechanism of photosynthesis may resemble C4 plants. But CAM plants lack trans anatomy, chloroplast dimorphism, etc. And initial fixation in CAM plants is in night, and final fixation during daytime. Aim is to preserve water, and some examples for CAM plants for a familiarization: bryophyllum, calancho, sedum, opentia, pineapple, odd one question with the C3, C4, etc. C2, even CAM, odd one question. Calculation of ATP, etc. CAM is just a basic idea we should know. After that, 5 theory questions, 5 NEET model question with the key. That is regarding this episode. So what all things we learned, we can just pass through. Mainly 3 items: C2, C4, CAM, C2 synonym, then scientist, aim, enzymes, cell organs involved, drawback, etc. And this is the famous diagram for it, all these amino acid, etc. Then comes next cycle C4, that is avoiding the drawbacks of C2, Hagen slag, HSK, port shack, dicarboxylic acid cycle. Then again growing in hot condition with monocot dicot, typical example. And name C4 is due to first stable product. Double fixation will be there. Special type of anatomy called trans anatomy, two areas, mesophyll cell and bundle sheath cell. Enzyme is initial case, web case. First product is oxaloacetic acid 4 carbon. Then grana present in mesophyll cell, granule cell. Then we entered into bundle sheath cell. Features of bundle sheath cell, trans anatomy, adaptation, and in bundle sheath cell, grana absent, agranal chloroplast. After that, we entered into the reaction. For that, I will think it is better to show this diagram. Reaction. Then, after that, we entered into energy calculation in C4. Exam point of view important. Then we entered into a famous cycle, not in direct syllabus, CAM cycle, just for mentioning. Again, adaptation, Scott active, stomata. Example for CAM plans. After that, 5 theory question, 5 NEET model question with the key. Hoping that it is clear to you. If you like this episode, please subscribe all, like it, share it, comment on it. We can meet in the next episode. So thank you.